Hello, this is Zach Bird. I will be presenting Lab Report 1, Biomolecules. In this report, I will discuss biomolecules, their origins, their uses, and the important roles they play in homeostasis. What is a biomolecule? Biomolecules are substances produced by organisms that are essential for life. The four primary biomolecules are lipids, proteins, carbohydrates, nucleic acids. Where do we obtain biomolecules? We obtain biomolecules through the food we eat. We obtain lipids through food groups such as nuts, dairy, eggs, and meat products. Protein comes from dairy, nuts, legumes, meat products, eggs, and etc. Carbohydrates come from fruits, vegetables, grains, and various sugars. Nucleic acids come from fruits, vegetables, dairy, etc. Every organism contains a marginal amount of nucleic acids. How are monomers and polymers different? Monomers are molecules that can react with any other molecule to form a very large molecule, or polymer. Polymers are substances composed of macromolecules, which are composed of monomers. Protein, nucleic acid, and some carbohydrates are composed of polymers. Monomers are smaller molecules that eventually develop into polymers in the right circumstances. What monomers compose the primary biomolecules? Lipids are composed of fatty acids and glycerol. Proteins are composed of about 20 different amino acids joined together to form a chain. Carbohydrates are composed of glucose and fructose. Nucleic acids are composed of nucleotides. Compare and contrast lipids and carbohydrates. Similarities. Both lipids and carbohydrates are energy sources, carbon-based, and play a similar role in maintaining the structural integrity of a cell. Differences. Lipids provide long-term energy storage, while carbs offer short-term storage. Carbohydrates are soluble in water, but lipids are not. Both biomolecules have different metabolized energy yields, with lipids being higher by more than double. Biomolecule test results. What solutions did I use? I used Benedict's solution, Lugol's solution, Biorute's solution, and Sudan 3. How did I know I had a positive result? Benedict's solution turned orange when simple carbohydrates were detected in the solution. Lugol's solution turned black when starch was detected in the solution. Biorute's reagent turned purple when protein was detected in the solution. Sudan 3 began clumping when lipids were detected in the solution. What was the purpose of this experiment? In this experiment, we sought to discover the biomolecules included in a McDonald's Happy Meal. The results were unsurprising given the knowledge of biomolecular sources discussed in this class. My hypothesis. My hypothesis stated that if the McMush solution contained a specific biomolecule, the reagents would undergo a reaction. Procedures. Simple carbohydrates. To test for simple carbohydrates, you must first find or create a liquid solution to test. Step 1. Deposit 5 milliliters of your carb solution into a test tube. Step 2. Add 3 milliliters of Benedict's solution to the test tube. Step 3. Place a test tube into a safe container and boil for 5 minutes or until change occurs. Proteins. Similar to carbohydrates, to test for proteins, you must first create a liquid solution to test. Step 1. Deposit 5 milliliters of your solution into the test tube. Step 2. Deposit 10 droplets of Biorute's solution into the solution and wait for a color change to occur. Lipids. Find or create a lipid-based solution. Step 1. Deposit 5 milliliters of water and 5 milliliters of your solution into a test tube. Step 2. Deposit 5 drops of Sudan 3 into the test tube and wait to see if the solution diffuses. Results. The Benedict solution had a positive reaction with the hamburger bun, the french fries, the soda, and the apple slices, showing those items contain basic carbohydrates. The Lugol solution reacted positively with the hamburger bun and the french fries, proving those items contained starches. The Biorute's reagent reacted positively with the hamburger patty, proving it contained a substantial amount of protein. The Sudan 3 solution reacted positively with the hamburger patty, hamburger bun, and french fries, proving those items contained lipids. Discussion. Were there any outside factors that may have influenced the results? I do not believe so. Other than what I interpreted as someone drinking from a water bottle in the background briefly, there were no outside contaminants to my knowledge, and a sterile field was maintained. What portion of the children's Happy Meal provided the 4 biomolecules? Carbohydrates, hamburger bun, french fries, soda, apple slices. Starch, hamburger bun, french fries. Protein, hamburger patty. Lipids, hamburger patty, hamburger bun, french fries. Was my hypothesis supported by the experiment? Yes. In my hypothesis, I predicted the solutions would react with the solutions. Are there any further experiments that could be done based on my results? Yes, a future scientist may examine my results and test the additives in a Happy Meal to further distinguish its properties.